Our next group of organic compounds are lipids or fats. Lipids contain carbon, hydrogen, and oxygen, as well as carbs, but a little bit less of them, and sometimes phosphorus. Lipids are insoluble in water. Remember, if you try to mix oil and water, it doesn't happen. There are three main types of fats: neutral fats or triglycerides, phospholipids, and steroids. Let's talk about triglycerides first. Example of triglycerides is neutral fats. That is a solid animal fat. When you eat meat and you see fat in it, that would be triglycerides. Mostly they are made of three fatty acids bonded to a glycerol molecule. Okay, please don't let those names to intimidate you. All you need to remember is that triglycerides are made of a head, which is a glycerol, and three tails. Those three lines are tails. So if you need to describe a triglyceride structure, your answer would be one head called glycerol and three tails, which are fatty acid chains. Main functions of triglycerides: energy storage as a fat, and insulation and protection of the organs. For example, our kidneys are always surrounded with a layer of triglycerides, of fats, just to protect them and partially to keep them in place. There are two types of those fatty acids that make tails of triglycerides. Some of them are straight, some of them are slightly crooked. So straight ones are called saturated fatty acids, and they have maximum number of hydrogen. Those are your solid animal fats, and also butter. Unsaturated, that can be bonded once or usually many times, and they lack some hydrogen molecules. Those are unsaturated. They have reduced number of hydrogen, and those are your oils that are liquid at room temperature. The only exception would be a coconut oil that can be solid at room temperature, but coconut oil is unsaturated fat. Next group of fats are phospholipids. Imagine that you take that glycerol with three tails, which is your regular triglyceride. You remove one of the tails, one of the fatty acids, and replace it with what is called phosphate group. So what is the structure of phospholipids? Still glycerol, two fatty acids, not three, and one phosphate group. Or simply you can describe it like a head made of glycerol and phosphate group and two tails or fatty acids. Those heads and tails have different properties, and phospholipids, main function of them, they are important in cell membrane structure. Our cell membranes would not exist without phospholipids. Number 3, steroids. Steroids are 4-ring structures. That's all you need to memorize. 4 rings connected together. That's common for all steroids. And then a tail. The tails are different. What are examples of steroids? Cholesterol. Cholesterol is a component of our cell membranes. When its level is too high, it's not good, but we still need it for our membranes. Vitamin D would be steroid by structure. Some steroid hormones like testosterone, and also bile salts. There are also other lipids in the body. Those would be fat-soluble vitamins like A, E, and K. Those would be some of the chemicals like prostaglandins, lipoproteins, and I just mentioned them to let you know that there are other lipids in the body. What you need to know is 3 groups of lipids: triglycerides, phospholipids, and steroids. Our next group of organic molecules are proteins. Each protein is a very complex large molecule polymer which is made of building bricks, monomers, amino acids. What is amino acid? Amino acid is a complex molecule that always contains carbon, hydrogen, oxygen, nitrogen, and sometimes sulfur and phosphorus. And all of those amino acids have amino group, have carbon, have carboxyl group, and side chain. So those three, carboxyl group, carbon, and amino group, are common for absolutely all of the amino acids, and we have 20 types of them. Their side chains can vary. To make a protein out of amino acids, they should go through the dehydration synthesis. Remember, synthesis out of out of those two amino acids, we create one beginning of the chain of protein, and the bond connecting them would be called peptide bond. It's a type of strong covalent bonds. And since water is released during that reaction, it's called dehydration synthesis. So you have a question, what proteins are made of? They are made of amino acids through the reaction called dehydration synthesis, when amino acids are connected together using peptide bonds. And of course, there will be much more than two. There will be dozens, hundreds, sometimes thousands of those amino acids. Very often on your tests there are questions about protein structure. Primary structure of the protein, that is your chain of amino acids connected by peptide bonds. That chain should be very long, so we need to organize it somehow. The chain is a primary structure. When we're trying to organize it, we turn it into a spiral, and the links of that spiral will be connected with weak peptide bonds. Here are peptide bonds. That would be your secondary protein structure. Then we still need to organize that spiral or that sheet right here. And it will be a tertiary structure when the spiral gets folded. The folded spiral would be tertiary protein structure. Some proteins are even more complex than that. For example, this protein, hemoglobin, that we have in our blood to carry oxygen, it is made of 4 tertiary structures connected together. Those 4 folded chains. And that is called quaternary structure. So please know what each of those levels are. Protein structure is made of primary structure, it's a chain of amino acids. Secondary structure, that chain is folded into a spiral or just a sheet-like structure by hydrogen bonds. Tertiary structure, that spiral is folded. Quaternary structure, when we are using more than one folded tertiary structures. There are 2 major types of proteins. One of them, made of twisted spirals, is called fibrous or structural proteins. Here it is in the picture. They're usually long structures. And those are water insoluble, very stable. Examples in the body would be keratin in our nails and hair, elastin and collagen in our skin, and also in our tendons and ligaments, and certain contractile fibers in our muscles. So that's the shape. They are long. They are made of long protein spiral chains. Second type of proteins would be globular or functional proteins. Here is an example. They can have very different shape, but very often they look like a globe. So they are water soluble. They are very sensitive to environmental changes. It means they can be damaged when temperature rises. and they have specific active sites, so some other molecules can join them. Examples would be antibodies protecting us against bacteria and viruses, hormones for regulation, molecular chaperones, those would be for folding proteins, and enzymes. As you remember, enzymes are used to speed up chemical reactions. Now let's summarize structures of all the proteins. They are used for structure, to structure our body. Keratin, elastin, collagen make our muscles, make our tendons, ligaments. Also, collagen is in our bones, keratin is in our skin and nails. So that's structural proteins. Proteins are used for protection. Antibodies in our blood can destroy viruses and bacteria. They are used for regulation. Example would be hormones. They are used for digestion. Those digestive enzymes help to break our food into molecules. And finally, special enzymes, different types of enzymes, can speed up our body chemical reactions. Please know the protein structure, types of proteins, and protein functions.